Hello, everyone. This is Eunyoung Kim, and I'd like to present my research. The title is DeSIDE-DDI, Interpretable Prediction of Drug-Drug Interactions using Drug-Induced Gene Expressions. This is a table of contents for this presentation. Let me start with a brief introduction. Drug-drug interactions, also called as DDIs, mean a change in a drug's effect when the drug is taken together with another drug. It is important because these days it is common to take multiple drugs at once to reduce toxic effects but increase the efficacy. This is called as polypharmacy. But these polypharmacy strategies do not always result in good effects. They can also induce harmful outcomes, known as side effects. For example, NSAIDs are not encouraged to take with warfarin because of higher chance of gastrointestinal bleeding. To detect these dangerous drug combinations, there have been many attempts to build prediction models. Starting from this famous polypharmacy side effect prediction model, Decagon, many researchers utilized graph neural networks on various interaction networks. More on deep learning techniques, other omics data has been utilized. But still, there is a room to be improved in terms of interpretability and interactions with novel compounds. So in this research, I wanted to predict polypharmacy side effects using deep learning methods and drug-induced gene expression data for interpretability. This is the research overview. The model consists of two parts, feature generation model and DDI prediction model. Different datasets are preprocessed for each model. Given drug pair and side effect data, each drug feature is obtained using the feature generation model, and then drugs pass through neural networks with gated linear units to consider the co-administration effects. Finally, using latent representation of drugs and the side effect score is calculated. The model is evaluated considering 3 DDI cases, and it was validated with an independent dataset from DrugBank. To train the feature generation model, I used Link's L1000 data, which provides differential gene expression signatures when a certain compound is treated. After the following preprocessing step, I've got signatures for 19,156 compounds. On the other hand, for DDI prediction model, I used two-size database, which contains relations between drug pairs and side effects preprocessed from FDA adverse event reporting systems. The actual data was downloaded from BioSnap and processed to filter for side effects. The table shows the number of data before and after preprocessing. The last dataset is DrugBank for external validation. I downloaded 2 versions and preprocessed as two-size drugs. The table shows the number of drugs and drug pairs for each version, and unlike two-size, DrugBank data has one binary label for one drug pair. Now I will show you the model architectures. Feature generation model is a simple neural network. The model takes inputs as molecular fingerprints and compound physicochemical properties. Each feature passes separate neural nets and is concatenated to calculate the output. The purpose of this model is to generate drug-treated gene expressions for all compounds. Since small proportion of compounds were overlapped between links and two sites, not all compounds had expression signatures. To utilize all drugs in two sites, I decided to generate based on gold standard dataset. On the other hand, DDI prediction model is more complicated, consists of two key modules. The first is co-administration model, consisting of GLUs to control the information flow with the consideration of drug combination. The second is score calculation model. I applied translational embedding method to make two related drugs to be close in side effect spaces. And then the margin-based loss made scores positive and negatives to be distinguished. In detail, the co-administration model is developed to express co-administration effects to gene expression signatures. Here, the GLUs are used to control the information flow with the consideration of two drugs. It is a simple metric of sigmoid activation function that calculates each value to range between 0 and 1. Then it is multiplied as feature weights. This can help the model to distinguish drug pairs with the same drugs. This heat map shows how GLU affects the original features. Each row is drug feature of the same, but in combination with the other drug. Then the calculated drug latent representations and side effect embedding were put into the side effect score calculation module. Inspired by the knowledge graph embedding method, I applied translational embedding method to make two related drugs to be close in side effect spaces. The scoring function is defined as the distance between drugs considering both directions. Then the loss function is defined like this formula. With that, the model learns to make more gaps between scores of positives and scores of negatives. And this will be different for each side effect. In this study, there are mainly three cases of interactions that can happen. Unseen interactions, one unseen drug, both unseen drugs. Here, unseen means that the data was not used for training. Therefore, it is new to the prediction model. Unseen drug can represent newly found compounds in real world. Also, there is another point to consider, the process of external validation. Drug bank has DDI information, but the level of description is different. It describes at molecular level. Labels indicate the existence of DDIs. To handle this problem, I generated triplets by combining drug pairs with all side effects. Then I predicted and counted the number of positive side effects for each drug pair. Here, I assume that if the number of side effects is greater than the average number of side effects in the positive data set, the drug pair will have interactions. For example, if the threshold is 2, the drug pair of drug 1 and 2 is positive since the number of positives is 3 in this figure. Now, I will show you the results. The concentrated model showed good performances in all cases compared to previous studies. The model decagon was not able to predict interactions with new compounds, so the performance could not be obtained, and this INM model was implemented. I looked into specific side effect terms as some of you may be interested in these. The table shows the case 1 performance and the number of drug pairs in two sides for each DeSIDE related term. I further validated the predicted triplets in case 1. The model predicted around 19,000 negative triplets as positives, and in fact, the negatives are not real negatives. So I looked into those drug pairs in drug bank, and 13.97% were found. Also, among positively predicted triplets, I could find previously known fatal drug interactions, as listed in this table. Next is the external validation with drug bank. With the external validation strategy explained previously, I predicted all possible drug pairs in the old drug bank published in 2016. Among predicted positives, most were false positives. Then, the predicted pairs were searched on the new version published in 2020. The proportion of true positives increased by 35%, indicating that the model can capture potential interactions. Also, a permutation test was performed to validate false positives. The same number of false positives were randomly sampled and compared how many positives are included. Lastly, there are interactions of all cases mixed up in drug bank, so I checked how many of them are in each case in terms of two sides. I found that only 13% of validated drug pairs were case 1 and the others were case 2 and 3, which are more difficult prediction tasks. The external validation results show that the proposed model can capture DDIs of all cases. Moreover, a Bayesian study was conducted to examine which model is essential in predicting DDIs. The baseline is the original model and the model without feature generation model, and models without each co-administration model and score calculation model were compared. This result indicates that both co-administration model and the embedding module contributed the most to the model performance. The last result is feature analysis. I wanted to see how drug features change by the paired drugs. The heat map shows the changed features of the drug, cyclophosphamide, affected by each paired drug. Hierarchical clustering was done considering the correlation of features, and drug categories of each cluster were examined by drug ATC codes. Expression values of several genes were changed with co-administration. Different patterns were detected for each cluster. In more details, I focused on weighted genes. I searched on PharmGKB database for any known compound pathways. Then I found that weighted genes are found in each pathway of the drug pair, the cyclophosphamide and celecoxib. The highlighted genes were found in top weighted genes. In summary, I constructed deep learning-based prediction model for drug-drug interactions. I used drug-traded gene expression data to consider indirect interactions at transcriptomic level and applied the graph embedding technique to successfully train the model. Also, the simple GLU was adopted to calculate feature weights and mimic co-administration effects. This is a list of references, and I conducted this research under the advice of Professor Ho-Jung Nam, and the work was funded by the NRF grant. Also, I would like to give special thanks to my lab members. The proposed research has been published in Journal of Cheminformatics in March. Thank you for listening. Please contact me if you have any questions.